So the MagnetisMM-3 trial is an open-label, two-arm phase 2 trial evaluating the drug Elranatamab. This is a BCMA-CD3 bispecific T-cell engager. The idea is that it binds BCMA, which is almost exclusively expressed on plasma cells, and it brings in T cells and activates them through the CD3 portion of the molecule. The study, the compound was evaluated in a phase 1 study called the MagnetisM-1 study, and in the context of that study, responses were observed in a little over 60% of patients, and those that responded after a median of 9 months of follow-up, 77% continued to respond. So that supported further development of the compound in this phase 2 trial. The way the trial itself is designed is that myeloma patients with relapsed or refractory disease who have previously been exposed to a proteasome inhibitor, an immunomodulatory drug, and an anti-CD38 antibody, otherwise known as triple exposed, were eligible. The trial also allowed for patients who were ECOG-2 with platelet count less than 25, platelet count just greater than 25, and a creatinine clearance over 30. And was designed in 2 cohorts, a cohort of patients that were naive to prior BCMA-directed therapy, and a second cohort that had previously been treated with a BCMA-directed antibody-drug conjugate or CAR T-cell therapy. The trial is now fully accrued, and the data I'll be presenting at this meeting is an interim analysis from the BCMA naive cohort, inclusive of 94 patients whose data was available at the time of data cut on March 23rd, 2002. Patients are treated with a fixed dose of Elranatamab at 76 milligrams subcutaneously. And the primary endpoint is overall response by blinded independent investigator review, with secondary endpoints of duration of response, PFS, safety, and so on. The drug is given, as I said, subcutaneously through a 2-dose step-up schedule on day 1 and 4 of 12 milligrams and 32 milligrams to the target dose of 76 milligrams, with the 3rd dose on day 8. It's given weekly for the first 6 months among patients or 6 cycles on 28-day cycles. Among patients that are at the time of the 7th cycle have achieved greater than a PR for more than 2 months, the treatment interval increases to every 2 weeks with the 76 milligram subcutaneous dose. At this meeting, we will review the safety data for this compound. The main safety profile included hematologic toxicities for these patients, inclusive of anemia, neutropenia, thrombocytopenia events. The main non-hematologic toxicity was CRS. Other toxicities were fatigue, some GI toxicities, and so forth. In terms of the CRS, patients were required to be hospitalized for the first and second dose, so day 1 and 4 dosing. The first dose required a 48-hour hospitalization. The second dose, 24-hour hospitalization. In practice, most patients wound up being hospitalized just once and received both doses. The incidence of CRS across the 94 patients was approximately 60%, This did include 4 patients treated at a 44 milligram single step-up schedule, and that was later modified to this 2-step schedule that I mentioned. And with the 2-step-up schedule of 12 and 32 milligrams followed by 76, the incidence of CRS, particularly the incidence of grade 2 CRS, was significantly diminished to 17% of the population. There were no grade 3 CRS events. The overall response data so far is fairly early. The median follow-up at the time of the data cut was approximately 3 to 4 months. And the response was 60.6%. So approximately 61% of patients responded. The time to first response was 1.2 months. And of course, additional follow-up is necessary for evaluation of duration of response and best response and so forth. Well, I think, for patients now that have been previously exposed to an IMiD, a proteasome inhibitor, and an anti-CD38, particularly those that are refractory, as was the case, triple-class refractory patients in this study represented the majority of the patients. For those patients, overall survival outcomes are fairly poor. So having a response rate of 60% and what we know as based on the phase 1 trial, the MagnetisMM-1 data for responding patients is a fair bit of durability of response. This represents a major sort of advance in that patient population. These drugs will, and this data presented today supports further expansion and development of elranatamab as both a single agent and in combination in earlier lines of therapy. So I think initially elranatamab can enter the treatment space in the triple refractory or triple exposed patient population where it really fits an unmet need, and then in time, hopefully we will see data emerge that will reveal additional roles for elranatamab earlier in the treatment history of patients with multiple myeloma. For now, I think the MagnetisMM-3 study is designed as a registrational study. The current data presented at this meeting is for BCMA naive patients that have been triple exposed to 3 drugs, the 3 drug classes. This study does also include a BCMA pre-treated cohort, and as the data from this trial matures, the hope is that additional response will merit the sequential treatment with elranatamab following prior treatment with already approved BCMA antibody drug conjugates and BCMA-directed CAR T-cell therapies. I think one of the toxicities that we need to pay attention to and manage with this compound is the CRS signal. Now that, of course, actually is quite predictable with this drug. The majority of CRS events occurred within after the first dose, with sort of diminishing CRS events with second and very rare CRS events with third dose. So I think this is a toxicity that we as a community are getting more comfortable with managing, and will need to continue to evolve in terms of our management thereof to prevent CRS, not just for this drug, but for actually probably all bispecific T-cell engagers. Another I think side effect that appears to be emerging is the perhaps highlighted by the COVID-19 pandemic is the incidence of infections. So COVID-19 was infections occurred in, or COVID-19 related events occurred in approximately 15% of the patients reported. other infections such as CMV infection and PJP infection are also things that need to be attended to and prophylaxed against when possible. And I think the most important thing to highlight is that, the bispecific T-cell engagers, particularly those targeting BCMA, but also other targets are really, the cutting-edge therapies in myeloma, and we're all very excited about these off-the-shelf sort of available treatments that have such a high rate of efficacy among patients who have otherwise limited treatment options currently. So I think that this is a very good thing overall for our patients.